Good day, dear students. I am Chmidonov Sergei Nikolaevich, the head of Human Anatomy Department in Samara State Medical University. Today we'll talk about brain, telencephalon or end brain. In details, we'll talk about cortex, about surfaces, about ventricles, and also about hidden great matter which calls nuclei, basal ganglia. First, I want to tell you that that end brain consists of two hemispheres. You may see these hemispheres here from superior view on left picture and from inferior view on right picture. You also may see cortex, cerebri, vessels, and also you may see cerebellum, brainstem, and other parts. So, on this picture you may see the telencephalon from lateral view, and in details you may see gyri and sulci of superior lateral surface of telencephalon. So each hemisphere is composed of the pallium or rhinencephalon. You may see rhinencephalon here on this picture, and also you may see rhinencephalon in violet color here. You may read this term on this picture too. So next part is basal nuclei or basal ganglia. You may see in him drawing basal nuclei here, and also you may see in horizontal section of brain basal nuclei here under my pointer. Also you may see on this picture the parts of ventricles. Lateral ventricles are the cavities of hemispheres or end brain. And also next part of cerebrum of telencephalon is cerebral cortex, the youngest part, or cerebromental you may read. Also the youngest part in phylogenetically light of end brain. Also on this picture you may see the gyri and sulci or grooves of cerebral cortex. So the forebrain from which the in-brain arises originate initially in association with the olfactory receptor, and also you may see rhinencephalon and olfactory bulbi, and later becomes the organ controlling the animal's behavior. This organ consists of centers of instinctive behavior based on species reactions, conditioned reflexes, and also subcortical ganglia, the centers of individual behavior based on individual experience, conditioned reflexes, the cerebral cortex. According to these, the following groups of centers are distinguished in the telencephalon in the order of historical or phylogenetical development. The part of the brain concerned with the function of olfaction, the rhinencephalon, you may see also again in violet color on this picture of these parts, rhinencephalon. It's the oldest, I repeat, part, at the same time the smallest part, which located ventrally. You may see it here. The basal or central ganglia or nuclei of the hemispheres or the subcortex is the out part of the end brain. It calls also palencephalon, hidden in the depth. And the cortex composed of gray matter, the youngest part, the neoencephalon, at the same time the largest part covering all the other parts like a cloak of mantle, hence its other name, the pallium in Latin. In addition to the two forms of behavior recorded in animals, a third form develops in man. This is collective behavior based on the experience of the human collective form, the process of the labor activity of man, and intercourse of humans by means of speech. It's very important. Speech, this form of behavior, is linked with the development of the youngest surface layers of the cerebral cortex composing the material substance of the second synalic system or verbal system. So, in the process of evolution, the in-brain develops most rapidly and intensively among all parts of the central nervous system. In man, it becomes the largest part of the brain and acquires the shape of two large hemispheres. Two large hemispheres. You may see this hemisphere from superior view. So, right and left. All these parts connected between each of them with the corpus callosum, the greatest chemisura in central nervous system. And you may see the parts of this corpus callosum in number 9. Lamina terminalis, rostrum, genu, truncus, and splenium. Let's in details we'll talk about cortex and youngest part of cerebrum. So, three surfaces are distinguished in each hemisphere: superior lateral, medial, and inferior here. Each hemisphere has anterior pole or frontal pole, polus frontalis, occipital pole or posterior pole or polus occipitalis, and also temporal pole or polus temporalis. So you may see the parts of hemisphere here. In a red color, you may see from superior view frontal lobe. In green color, you may see parietal lobe. In blue color, you may see occipital lobe. So in yellow color, you may see the temporal lobe. So the surface of hemisphere, the pallium, is formed by a regular layer of gray matter containing nerve cells. This layer also called the cerebral cortex or cortex cerebri in Latin. It seems to be laid in folds, as a result of which the surface of the pallium has a very complicated pattern form of sulci alternating in different directions with the elevations called gyri. The size and shape of the sulci are marked by the considerable individual variability, as a consequence of which not only the brain of different person but even the hemispheres of a given person are not quite similar in the pattern of the sulci cerebri. You may see the grooves of this surface, which called sulci. The deep and constantly present sulci used as guides to divide each hemisphere into large areas called lobes, lobi, as I told you. These are in turn separated into small lobes and gyri. Each hemisphere has 5 lobes. I showed you 4 only: frontal, green, frontal in red. In green color you may see parietal lobe, in yellow color temporal lobe, and in blue color occipital lobe. And also in each hemisphere is situated a small lobe, the insula, hidden in the depth of the lateral sulcus here. So this superior lateral surface, you may see this surface here on this picture, is separated in two lobes by three sulci. Lateral sulcus between green part, red part and yellow part. So the lateral sulcus or Sylvian sulcus is situated between frontal, parietal and temporal lobes. Next you may see the central sulcus in number 13 between red and green colors, between frontal and parietal lobes. And also you may see the part of third sulcus here, which begins on the medial surface. Here you may see number 14, in number 14. And so it passes on the superior lateral surface, particularly you may see it here, and divided two lobes, parietal lobe from occipital lobe, green from blue. So the central of Aranda sulcus, you may see, rises on the superior border of the hemisphere slightly to the back of its middle part and passes forward and downward. The area of the hemisphere in front of the central sulcus is related to the frontal lobe. Part of the brain surface to the back of the central sulcus in green color composes the parietal lobe, which is separated from the temporal lobe in yellow color, lying below by the posterior part of the lateral sulcus. The posterior boundary of the parietal lobe is formed by the end of the above mentioned parieto-occipital sulcus. Here, sulcus parieto-occipital is located on the medial surface in number 14 of the hemisphere. This boundary, however, is incomplete, because the parieto-occipital sulcus does not stretch far on the superior lateral surface, as a result of which the parietal lobe is directly continuous with occipital lobe. You may see the continuation parietal lobe to occipital lobe. So the latter also has no clearly defined boundary separating it from the temporal lobe situated in front. In view of this, the boundary between these lobes is drawn typically by means of a line passing from the parieto-occipital sulcus to the inferior border of hemisphere. Each lobe consists of number of gyri, which are called lobules, at places bounded by the sulci of the brain surface. The frontal lobe. At the posterior part of the outer surface, or superior lateral surface of the lobe, this is precentral sulcus. You may see on this picture number 14. Sulcus precentralis, stretching almost parallel to the central sulcus. It gives rise to two longitudinal sulci, the superior and inferior frontal sulci. You may see these frontal, superior and inferior in number 16 sulci. Sulcus frontalis superior and sulcus frontalis inferior, it's in Latin. As a result, the frontal lobe is divided into 4 gyri, 1 vertical and 3 horizontal. The vertical precentral gyri number 1, you may see here, is between the central and precentral sulci. The horizontal gyri of the frontal lobe are as follows: superior, middle in number 8, and inferior. So the superior frontal gyrus passes above the superior frontal sulcus, parallel to the superior border of the hemisphere and onto its medial surface. The medial frontal gyrus stretches between the superior and inferior frontal sulci, and the inferior frontal gyrus located between the inferior frontal and lateral sulci. Branches of the lateral sulcus, which project into the inferior frontal gyrus, divide the last named into three parts. You may see these three parts here. A posterior part, pars articularis, between the lower end of the precentral sulcus and the horizontal ascending ramus of the lateral sulcus. Next, triangular part, between both rami of lateral sulcus. And finally, the orbital part, in front of the horizontal anterior ramus of the lateral sulcus. Three parts, I repeat: apercularis, triangularis, and orbitalis. Parietal lobe in green color. On this lobe, almost parallel to the central sulcus, you may see the central sulcus again. This is past central sulcus. You may see under my pointer. Sulcus postcentralis, which usually blends with the intraparietal sulcus. You may see this blending here. Sulcus intraparietalis in Latin. And you may see that this sulcus goes horizontally. According to the position of this sulci, the parietal lobe is separated into one vertical and two horizontal gyri. The vertical, past central gyrus in number 4, gyrus postcentralis in Latin, passes behind the central sulcus in the same direction as the precentral gyrus in frontal lobe. Above the intraparietal sulcus is the superior parietal gyrus in number 9, or lobulus parietalis superior in Latin, which extends also to the medial surface of the hemispheres. You may see this part here. Below the interparietal sulcus lies the inferior parietal lobe in number 10, lobulus parietalis inferior, which passes to the back, curves around the ends of the lateral sulcus and the superior temporal sulcus, and is last in the occipital lobe. You may see it here. The anterior part of the inferior parietal lobule, bending around the lateral sulcus, is called the gyrus supramarginalis. You may see this gyrus here. The middle part, curving around the superior temporal sulcus, is known as gyrus angularis, and you may see this part here under my pointer. The temporal lobe, the lateral surface of temporal lobe in the yellow color, you may see on this picture, has three longitudinal gyri separated from one another by the superior in number 18 superior and inferior sulci in number 19. The superior temporal gyrus, gyrus temporalis superior in number 5, is between the lateral and the superior temporal sulci. Its superior surface, hidden deep in the lateral sulcus, has three short transverse temporal gyri, gyri temporalis transversi. You may see these gyri here, 1, 2, and 3, or Heschle convolutions. Hesher is the surname of anatomist. Between these superior and inferior temporal sulci stretch the middle temporal gyrus, gyrus temporalis medius in number 6, middle temporal gyrus, gyrus temporalis medius, below the last named and separate from it by the inferior temporal sulcus in number 19 is the inferior temporal gyrus in number 7, gyrus temporalis inferior, forming the junction between the lateral and the inferior surfaces of the temporal lobe. The occipital lobe. You may see this lobe in blue color. The sulci of the lateral surface of this lobe are variable and inconstant. The transverse occipital sulcus, sulcus occipitalis transversus, is distinguished among them. It's usually joined to the end of the interparietal sulcus. So you may see the medial surface of hemisphere here. The medial surface of hemisphere carries the callosal sulcus. You may see corpus callosum in number 9 and callosal sulcus in number 12. So the callosal sulcus passes directly above the corpus callosum and it continues posterior with the deep hippocampal sulcus, running forward and downward. Here. Between gray and gray color on this picture and violet color, this one. So parallel to and above the callosal sulcus extends the sulcus singuli. You may see this sulcus in number 13 on this picture. Sulcus singuli, which arises in front under the rostrum here. And passes to the back, its posterior and terminating on the superior border of hemisphere. The space between this hemispheric border and the sulcus singuli is related to the frontal lobe, to the superior frontal gyrus. A small area above the sulcus singuli bounded by its posterior end at the back and by a small paracentral sulcus, sulcus paracentralis, in front is called paracentral lobe, lobulus paracentralis. You may see this lobe here in number 4. So it corresponds to the medial surface of the superior end's central gyri, where continues one with the other. To the back of the paracentral lobe, its quadrangular surface, the precuneus, you may see in green color, bounded anteriorly by the end of the sulcus singuli. You also may see the end of sulcus singuli here. Inferiorly, the small subparietal sulcus, and posteriorly by the deep parieto-occipital sulcus. You may see parieto-occipital sulcus here in number 14. This is the border between parietal and occipital lobes. The precuneus belongs to the parietal lobe, as I told you, in green color. Behind its sharply demarcated area of cortex related to the frontal lobe, this is cuneus, bounded anteriorly by the parieto-occipital and posteriorly the calcarine sulcus. You may see the calcarine sulcus in number 15, which converge at angle. Inferiorly and posteriorly, the cuneus is a contact with the medial occipital-temporal gyrus. Medial occipital-temporal gyrus. You may see this gyrus in number 7 passes and to number 8. From number 7 to number 8. Between the sulcus singly and the callosal sulcus stretches the gyrus singly. You may see in number 1 this gyrus, which by means the isthmus is continuous with the parahypocarpal gyrus terminating as a hook-like structure, the uncus. You may see in number 2 a parahypocarpal gyrus, and it passes to uncus, like hook. The hippocampal gyrus is bounded on one side by the hippocampal sulcus, carrying around brain stem, and no the other side by the collateral sulcus and its anterior continuation called the renal sulcus. Renal sulcus. The isthmus are a constrict part of the junction of the gyrus singly and the hippocampal gyrus. You may see the isthmus here under my pointer. So the isthmus is behind the splenium of corporis callosum, at the end of the sulcus, formed at the fusion of the parietal occipital and calcarine sulci. You may see parietal occipital and calcarine sulci here. The gyrus singly, isthmus, and hippocampal gyrus constitute the gyrus fornicatus, describing almost a complete circle open only below and in front. The gyrus fornicatus is not related to any of the lobes of the cortex. So, the margins of the hippocampal sulcus, one can see a narrow, denticulate, gray band which rudimentarily dentate gyrus. So you may see this gyrus here symmetrically. So next we'll talk about inferior surface. You may see this part and this surface here. Its anterior part is related to the frontal lobe in red color in number 2. Here, parallel to the medial border of the hemisphere, passes the olfactory sulcus. You may see in number 7 olfactory sulcus, enlarging the olfactory bulb and tract. And number 1 you may see olfactory bulb, and it passes to olfactory tract. Between the sulcus and the medial border of hemisphere, this one under my pointer, this is gyrus rectus, in continuation of the superior frontal gyrus. Several inconstant orbital sulci in number 2 are seen on the inferior surface lateral to the olfactory sulcus. You may see that they are inconstant here. So orbital gyri can be regarded as continuation of the middle and inferior frontal gyri. The posterior part of the basal hemispheric surface is formed by the inferior surface of the temporal and occipital lobes, whose boundaries are unclearly defined. Here, 2 sulci are seen on this area. The occipito-temporal sulcus, you may see this sulcus here, passing from the occipital pole You may see occipital lobe in blue color to temporal lobe. And also you may see collateral sulcus extended parallel to it. The sulcus is continuous entirely with the renal sulcus. Between them runs the lateral occipital-temporal gyrus occipitotemporalis. Two gyri are located medial to the collateral sulcus, the medial or lingual sulcus, medial occipitotemporal gyrus. So the hippocampal gyrus or parahippocampal gyrus in number 5 between the anterior end of the collateral sulcus and the renal sulcus on the side, and the hippocampal sulcus covering around the brain stem on the other side. The hippocampal gyrus or parahippocampal gyrus, being adjacent to the brain stem, is one on medial surface of the hemisphere. So let's in details we'll talk about structure of rhinencephalon or olfactory brain. So rhinencephalon is phylogenetically the oldest part of the forebrain. It arises in association with the olfactory receptor when the forebrain has still not become the organ that controls the animal's behavior. All its components are therefore different parts of the olfactory analyzer. The concept of analyzer. In fish, almost the whole forebrain is an olfactory organ. With the development of the new cortex encountered in mammals and man, a new part of forebrain develops. As I told you, pallium or cortex or neocortex. But it also goes through a long developmental course and contains 3 parts differing in phylogenetic age. The oldest part are as follows: paleopallium, archipallium, neopallium is new part. So let's in detail we talk about the oldest part, rhinencephalon. And you may see 2 parts of rhinencephalon, peripheral part, and next we'll talk about central part. You may see these parts here: olfactory bulb, olfactory tract, olfactory triangle, anterior perforated substance, stria diagonalis, olfactory tubercle, medial and lateral olfactory stria. And so you may see in yellow color olfactory bulb, olfactory tract, olfactory triangle with stria, olfactory tubercle, and anterior perforated substance here. And next you may see these parts here too. You may see olfactory bulb, olfactory tract, olfactory triangles here, stria, lateral and medial, olfactory tubercle, and anterior perforated substance. Also you may see on this preparation these parts. Next we will talk about central part of olfactory brain, the oldest part of our brain in phylogenetically light. Fornix gyrus, hippocampus, dentate gyrus, uncus or hook, interlimbic gyrus, fascicular gyrus, and also indusium, griseum over the corpus callosum. So you may see and read these parts here. They are central part of olfactory brain or rhinencephalon, and you may see this part in brain. You may see hippocampus, gyrus dentatus, fasciolaris, gyrus fasciolaris. You may see in details these parts here. Gyrus dentatus goes posteriorly and passes to fascicular gyrus, and fascicular gyrus covered with gray matter this part of cerebrum, white matter, which cause corpus callosum and form indusium griseum over the corpus callosum. And you may see hippocampus on this transverse section. And you may see that it forms the lateral border or lateral wall of cornu inferior of lateral ventricle. This is cavity of end brain. So you may see the central part of rhinencephalon in violet color here, and I told you about this part. You may see area subcallosa, gyrus singly. It passes posteriorly and it goes downward and passes to isthmus, and isthmus passes to gyrus parahippocampalis and uncus. And all these parts form gyrus fornicatus. So in yellow color you may see gyrus dentatus, which passes to gyrus fasciolaris, and as I told you, it goes upward and covers the corpus callosum. You may see indusium griseum. This is the thinnest part of gray matter which cover the corpus callosum. So next we'll talk about basal nuclei, basal ganglia in details, and you may see these ganglia here. You may read these ganglia. So there claustrum, amygdaloid body, lentiform nucleus, caudate nucleus. So in addition to the gray cortex of the surface of the hemisphere, masses of gray matter are present in the depth of its tissue. These are called basocentral or subcortical nuclei, or subcortex. As distinct from the cortex, which has the structure of screen centers, this subcortical nuclei possesses the structure of nuclear centers. So you may see these centers here. Corpus striatum consists of two parts: the caudate nucleus and a part of lentiform nucleus, putamen. And so you may see globus pallidum, which consists of two parts, medial and lateral stria. So you may see these parts here. Let's in detail we see these parts in this horizontal section. I point you the part of striatum. This one is nucleus caudatus. Nucleus caudatus. This is the head of nucleus caudatus, and this part, little part, is tail of nucleus caudatus. Cauda in Latin is the tail. So you may see a lentiform nucleus, which consists of three parts. This lateral part is called putamen, putamen, and these two parts are called globus pallidum, medial and lateral stria. And laterally you may see a thin part of gray matter over the putamen. This is claustrum. But we cannot see on this section the amygdaloid body, and we will see on next picture. So in detail you may see striatum. Let's in detail talk with this picture about this part of brain. The caudate nucleus lies above and medial to the lentiform nucleus, and is separated from it by a layer of white matter called the internal capsule. You may see this white matter here. This is internal capsule. Capsula interna in Latin. The thickened anterior part of the nucleus is head. You may see in number 2 in this picture. Caput nuclei caudati. Forms the lateral wall of the anterior horn of the lateral ventricle. You may see this cavity and the inferior lateral border or wall of this ventricle, while its attenuate parts, the body and tail, corpus and cauda nuclei caudati, number 3 and in number 4, you may see these parts here. So stretch to the back on the floor of the central part of the lateral ventricle. The tail curves on the superior wall of the inferior horn. You may see this little part of tail here, and also you may see inferior horn here of ventricle. Medially, the caudate nucleus adjoins the thalamus. You may see thalamus. I repeat that this part is diencephalon. So from which it's separated with a band of white matter called the stria semicircularis, stria terminalis. Anteriorly and inferiorly, the head of the nucleus approaches the anterior perforated substance, where it's united with the lentiform nucleus, with the part called putamen. In addition to this white union of these two nuclei of the ventral side, there are fine bands of gray matter. You may see these bands of gray matter in number 14 here. These bands, alternating with the white bundles of the internal capsule, are responsible for the name corpus striatum, and I told you about this earlier. The lentiform nucleus lies lateral to the caudate nucleus and the thalamus, and you may see this nucleus here. On the horizontal section through the hemisphere, the medial surface of the lentiform nucleus facing the internal capsule has the shape of an angle shaped here, whose apex is directed centrally. The anterior side is parallel to the caudate nucleus, and the posterior side is parallel to the thalamus. The lateral surface of the nucleus is slightly convex. You may see this convection, and is directed at the lateral side of the hemisphere in the region of the insula. You may see this hidden part of the cortex, which are called insula. Anteriorly and ventrally, the lentiform nucleus is fused with the head of caudate nucleus. On the frontal section, the lentiform nucleus is wedge-shaped. The apex of the wedge is directed medially and the base laterally. Two parallel white layers called the medullary lamina You may see this lamina here. Divided the lenticular nucleus into three segments. Lateral gray segments called putamen, this one, and you may see on this left picture in 11 number. And two medial lighter colored segments united under the term globus pallidum. You may see these two parts in number 12. All these parts in blue color are globus pallidum. Also, you may see these parts here on this horizontal section. Globus pallidus has a distinctive macroscopic appearance and also differs from the other parts of the corpus striatum histologically. It's phylogenetically older than putamen. This is paleostriatum, and the putamen of a caudate nucleus is neostriatum. In view of these features, the globus pallidus is now distinguished as a specific morphological structure, the palleum, while the term striatum is used as designation only for the putamen and caudate nucleus. The term lentiform nucleus loses its initial meaning, and it should be used only in purely topographical sense. The caudate and lenticular nuclei are now called striopallidal system instead of corpus striatum. You may see in number 3 and number 11 and pallidum. This system is a principal part of extrapyramidal system, and besides, it is a higher center of control of vegetative functions concerned with the thermoregulation and carbohydrate metabolism, and predominates our similar vegetative center of the hypothalamus. Next, we will talk about claustrum. You may see this part here on this horizontal section. So, claustrum is thin sheet of gray matter in the region of the insula. You may see insular cortex here, between it and putamen. It's separated from the putamen by a thin layer of white matter. You may see this white matter here, and it calls external capsule. And it's separated from cortex of insula with thin white matter, which called extrema capsule. So, certain authors do not relate the claustrum to the basal ganglia, but consider it to be deep layer of the insular cortex, which became separated with the development of the capsule extrema. According to another point of view, the claustrum is absolutely a dependent structure related to the basal ganglia in origin. So, on this picture number 7, you may see amygdaloid nucleus, corpus amygdaloidum, or epistriatum. You may see also on this picture and on this picture in number 13. Amygdaloid nucleus or epistriatum lies under the putamen in the anterior end of the temporal lobe. It does not reach the temporal lobe, but lies in front on the apex of the inferior horn of lateral ventricle. Morphologically, the amygdaloid nucleus is a posterior ventral continuation of the claustrum. It's evidently a subcortical olfactory center where a bundle of fibers passing from the olfactory lobe and the anterior perforated substance terminates. So, next we'll talk about cerebral cortex in details. You may see this photograph. This is Einstein cortex, Einstein brain. So you may see these parts of cortex, paleocortex, paleocortex, or paleopallium, a small cortical area on the ventral surface of frontal lobe lying close to the olfactory bulb. It includes olfactory tubercle and its surrounding cortex, including a section of the interior perforated substance. You may see this part here. Next, you may see archipallium or archaeocortex. This is component of the temporal lobe. At first it was on the lateral surface of the hemisphere, but later, under the effect of intensively growing neopallium, it curved up to form a sausage-like structure, the hippocampus or Ammon horn, and migrated medially into a lateral ventricle of the brain as a projection of the inferior horn. The hippocampus is covered by cortex, archicortex. So you also may see the dentate gyrus and hippocamp here. Next, you may see the middle cortex or mesocortex, which divided in two zones. One separates the new cortex from the archicortex, periarchicortic zone, the other from the paleocortex, paleocortical zone. These zones occupy the lower part of the insular lobe, the parahippocampal gyrus. And at last, the new cortex or neopallium. So a neocortex in which the higher centers have appeared. So neocortex in humans is the most significant in land, accounting for approximately 96% of the entire surface of the cerebral hemispheres and is characterized by multi-layering. You may see 6 layers of neopallium or neocortex, and also you may see these layers on this histological section. Also you may see the gyri and sulci of cerebrum. You may see the cortex, neocortex, multi-layer part of cortex, and also you may see white matter here. So neocortex organization you may see on this picture. There are 2 basic elements, brain center and scattered elements. Brain center of the cortical end, you may see the cortical end of the analyzer, has not strictly defined boundaries but consists of the nuclear and scattered parts. The core or nucleus of cortex represents in detailed and accurate projection the cortex of all elements of a peripheral receptor and is necessary for the implementation of higher analysis and synthesis. And scattered elements are located on the periphery of nucleus and can be scattered far from it. They carry out simpler and more elementary analysis and synthesis. And also you may see cortex on this picture. On this picture you may see in details insula part. Insula. So let's we talk about insula. This lobule becomes visible when the margins of the lateral sulcus which overhang it are drawn apart or removed. You may see this removed part here, and in details you may see the insula. These margins are related to the frontal, parietal and temporal lobes. and are termed operculum. The insula is triangular, you may see triangular shape of insula, and its apex is directed forward and downward. This one. Anteriorly, superiorly, and posteriorly, the insula is separated from the adjoining parts by a deep circular sulcus. The surface of the insula is covered by several short gyri. And you may see these short gyri here. So you may see the projection of analyzers here, motor and sensor parts on precentral and postcentral gyros. You may see that the motor parts have projection from face, tongue and hand. You may see this projection here, and also you may see this projection here of motor cortex. But you may see that little part of cortex has projection from legs, foot and our body, little part. But the greatest part is from hand, face muscle, mimic muscle and tongue. So you may see this projection in three-dimension model. So you may see the projections of functional zones on cortex. In number 1 you may see motor zone of cortex in green color. In number 2 the core of skin analyzer. You may see this part on parietal lobe, postcentral gyrus and area or lobulus parietalis superior parietal lobulus. So in number 3 you may see in red color the center of targeted combined movements on the inferior parietal lobulus. Next, in number 4 you may see the verbal system or second signal system, part of second signal system in brown color, visual analyzer or written language. When we read the book and we understand the text. So this part is called angular gyrus. In number 5 you may see auditory analyzer of oral speech. It's also a part of verbal system or second signal system. This is supramarginal gyrus. In number 7 you may see the part of temporal lobe and projection of auditory analyzer, hearing analyzer. So in number 8 a part of taste analyzer. Number 10 motor analyzer of combined rotations of the head and eyes. In number 9 a motor analyzer of oral speech, and in number 11 a motor analyzer of written language, to write the words. So number 9, number 11, number 4 and number 5, all these 4 centers are below to second signal system, verbal system. And you know all these systems are write system, read system, to understanding speech, to understanding written speech, written text, and also oral speech and hearing. Oral speech motor center is in number 9. The posterior part of inferior frontal gyrus, and sensoric sensitive center of oral speech, of hearing, is in number 5, supramarginal gyrus. And also you may see the centers of cortex on the medial part, on the medial surface. You may see on the occipital lobe the center of visual analyzer in number 3, and number 4 the center of stereognostic feeling, the core of skin analyzer, which passing from lateral surface, and also in red color in number 1 and 2 you may see 2 analyzers. This is parahippocampal gyrus, and it contains 2 analyzers, taste and olfactory analyzers. So you may see the disorders of writing, disorders of center of writing. As I told you, the center is formed in posterior part of medial frontal lobes, gyrus of frontal lobe in number 11. And you may see on this picture disorders of this center. And in details we will talk about lateral ventricles. You may see these cavities of and brain of Telencephalon. So 2 lateral ventricles are situated below the level of the corpus callosum, on either side of the midline in the hemispheres, as remnants of the primary cavities of both the ventricles of the brain. The whole thickness of the brain matter separates them from the superior lateral surface of the hemispheres. The cavity of each lateral ventricle corresponds to the shape of hemisphere. It begins in frontal lobe as anterior horn. You may see frontal lobe and anterior horn, covering downward and laterally, then its central part. Central part stretched through the parietal lobe. At the level of the posterior margin of the corpus callosum turns downward and passes forward in the temporal lobe as inferior horn. So the distending part of the ventricle gives out the projection to the back into occipital lobe. This is posterior horn. The medial wall of the anterior horn is formed by a septum pellucidum. You may see this part here, septum pellucidum, under my pointer. It separates it from the similar horn, right from left, on other hemisphere. The lateral wall and a part of the floor of the anterior horn are occupied by a gray projection, the head of caudate nucleus, caput nuclei caudati, while the superior wall is formed by the fibers of the corpus callosum. The roof of the central narrowest part of the lateral ventricle is also composed of fibers of the corpus callosum, whereas the floor is formed by the body of the caudate nucleus, corpus nuclei caudati, and part of the superior surface of the thalamus. The posterior horn is surrounded by a layer of white nerve fibers arising from the corpus callosum. This layer is called tapetum or carpet. The medial wall of the posterior horn carries on elevation the calcar avis. You may see number 11 here, calcar avis, formed by projection of a calcarine sulcus which stretch on the medial surface of the hemisphere. The superior lateral wall of the inferior horn is formed by the tapetum, which is a continuation of the similar structure surrounding the posterior horn. The thinned part of the caudate nucleus, its tail, carrying downward and forward, passes on the superior wall of the inferior horn from the medial side. A wide projection, the hippocampus in number 4, or a mons horn, extends for the whole length of the medial wall of the inferior horn. It's produced because the hippocampal sinus presses in deeply from the outside. Small sulci divide the anterior end of the hippocampus into several small projections. The middle border of the hippocampus carries a triangle fimbria hippocampi. You may see this fimbria here, which is a continuation of the posterior column of the fornix, crus fornicis. On the floor of the inferior horn is an elevation, the collateral eminence, produced by the collateral sinus pressing in from the outside. On the medial side of the lateral ventricle, its central part and inferior horn is a projection of the pia mater, forming here vascular plexus, the chorioid plexus. You may see this plexus here in violet color. So the plexus is covered by epithelium, which is a rudiment of the undeveloped medial wall of the ventricle. The choroid plexus of the lateral ventricle is the lateral margin of telechorioida of third ventricle, and it produces the liquor or cerebral fluid. And at last of our lecture, we'll tell some words about limbic system. In details, you will study this system on physiology. The limbic system is a functional union of brain structures involve the organization of emotional, motivational behavior, food, sexual, defensive instincts, wake-sleep cycle. So, morphofunctional organization of limbic system you may see on this picture. First complex is antecortex, olfactory bulbs, olfactory tubercle, septum pellucidum. Second complex is paleocortex, dentate fascia, cingulate gyrus. And the third complex is the structure of the insular cortex and parahippocampal gyrus. Subcortical structures, amygdaloid bodies belong to limbic system, yeah? A nuclei of the septum pellucidum, anterothalamic nucleus, and mammillary bodies, reticular formation. And you may see this part of limbic system which form our behavior, food instinct, sexual instinct, and other instincts here. So also you may see on this picture the parts of limbic system. All these parts belong to Telencephalon, which you can see here. But also you know that there are not only parts of Telencephalon, because thalamic nucleus belong to thalamus, diencephalon, mammillary bodies belong to hypothalamus. Hypothalamus is a part of diencephalon too. So you may see this part of limbic system here. So the lecture is over. Thank you very much for your attention. Goodbye.